So the next section is bioaccessibility study. And I think we go back to Patty for a overview. I think
We're in section 3 of your binder.
Yes. tab 3, Lauren promises this time.
So this section will discuss both. First section will be the physiology and composition of biofluids that we're proposing and modify since the last meeting. And also the second section will provide more detail on the experimental setup. I'll pass it to Dr. Jocelyn Clough from OEHHA.
Okay, thank you. So this section will cover, first we'll talk about the proposed biofluid compositions that we'll use. Then we'll define what is the bioaccessible concentration. Sorry. Okay. Then we'll talk about the traditional method that's typically used to measure the bioaccessible concentration. We'll talk about a dynamic measurement method the lab is using, and then we'll have some illustrative results at the end of the talk. So first we'll start by defining bioaccessibility. So bioaccessibility is the total amount of a substance in an exposure medium that's potentially available for absorption into the body. For this study, we're going to be talking about crumb rubber, air, dust particles associated with crumb rubber. And this little illustration on the bottom shows where chemicals can potentially be located on crumb rubber. So we can have chemicals on the outside that are available for absorption, and as chemicals move, as crumb rubber moves through the body and crumb rubber could possibly be degraded, you can have chemicals migrating from the inside to the outside and become available for absorption. So this slide's just a brief reminder of the exposure pathways we're going to be talking about. On the left, you can see the different components of the synthetic turf. We can measure the levels of chemicals in the components and the levels that you're exposed to, but like was mentioned earlier, risk is going to be determined by what's available, the types and amounts of chemicals that can get into the body. So at the last meeting, we received advice from panel members on the compositions of our biofluids. So we were advised that we could improve our gastrointestinal and dermal biofluids by adding lipids to the compositions. And also, it was suggested that we could consider, for the inhalation exposure, adding an additional compartment that would represent the phagolysosomal environment of macrophages in the lung. So what we've done, we've looked to the literature and tried to incorporate this advice and made changes to our biofluids. So just overall, what we've done is we've added relevant lipid components to both the gastric and intestinal artificial biofluids. We've also added a lipid skin component, and for inhalation, we've added a second biofluid, which represents the acidic environment within the macrophage. And in the next slides, I'll talk about each specific pathway and the biofluids we have for those. So first, we'll talk about dermal exposure. So dermal exposure is intended to simulate skin contact with crumb rubber, either directly or through resuspended particles in the air. On the left, we have a schematic of the structure of the skin. The layers are typically coated with a liquid layer that's a mixture of sweat and sebum, which help to protect it against the absorption of harmful chemicals. So to model this exposure pathway, we're going to use an artificial sweat biofluid. This is, oops. Sorry, spazzing. The sweat composition we propose is an aqueous composition. In our literature search, we found papers and studies that have shown that simple compositions can provide similar results to some of the more complex compositions. And to that, we're going to add this artificial sebum composition. This composition is composed of oils, lipids, and cholesterols, and we'll add it to the sweat to help mimic that surface layer. And the addition of this sebum component is expected to enhance the extraction of some of the more lipophilic chemicals that won't be extracted with just the simple sweat composition. So next for inhalation exposure. So this is intended to simulate the inhalation of crumb rubber particles in the air. So chemicals from inhaled particles can be absorbed in this first artificial biofluid, which is meant to represent the extracellular environment of the deep lung. This fluid, it's very inorganic and it does have low levels of lipids in it. The second artificial biofluid is what we've added, and it's meant to represent the alveolar phagolysosomal fluid within macrophages. These are resident immune cells within the lung that help protect it from the damage of particles that can get deep down. The composition is very acidic in contrast to the interstitial lung fluid that we propose. And it also has the addition of organic components, which can help to aid in the digestion of particles. So next for oral exposure, this is intended to mimic when crumb rubber particles are actually ingested. So we're going to use 3 different types of biofluids. So we're going to use saliva, gastric fluid, and intestinal fluid to simulate the absorption of chemicals from the GI tract. And on the left here, we have a little schematic of what the transport would look like. So first we'll talk about the saliva. So saliva is mostly aqueous fluid and it has roles in digestion. This is the component that the composition that we proposed at the last meeting. It's very simple and aqueous, and based on our search of the literature, we've chosen a new buffer that has the addition of important salivary enzymes. In our search, we were unable to find a validated composition that did have lipids in it, but based on the fact that the level of lipids is very low in saliva and the time crumb rubber is expected to spend in contact with this biofluid is very low, the overall concentration of chemicals extracted isn't expected to have a great impact on the total amount of chemicals that will be bioaccessible. So next we'll talk about gastric fluid. So the main role of gastric fluid is to start the breakdown of food and release nutrients. So this fluid it's very acidic and it has many enzymes. And so this is the fluid that we presented at the last meeting. It's very simple and has levels of enzymes, but on the left here we have the compositions that we're proposing based on our search. So we have one that represents a fasted condition and one that represents a fed condition. Based on the time since the last meal and the presence of food, this can have an effect on the pH of the gastric fluid, and it can also affect the types and amounts of chemicals that can be extracted, because as you can see with the fed composition, we have a milk component, which is intended to mimic, the standard macronutrient levels that is expected will be in a typical meal, such as proteins, carbohydrates, and fats. So you can notice the pH difference between the two fluids. The fasted composition is very acidic, while the fed composition has a higher pH. Compositions have been typically used in the pharmaceutical industry when they're testing the bioavailability of oral drugs. And typically they're large organic macromolecules, and we expect these compositions will be appropriate for this study. So after the stomach, the crumb rubber would pass into the intestine, where it's going to come into contact with intestinal fluid. This fluid typically further aids in digestion, and a majority of nutrient absorption occurs in the intestine. So this fluid has a composition that's similar to gastric fluid, but levels of certain constituents are different. Particularly bile and lipids are higher in the intestinal fluid, and the pH is also different from the gastric fluid. So our composition that we proposed at the last meeting was a simple sodium bicarbonate solution. These are our new proposed compositions. So they contain fat, and they also fat in the fed after a meal, and they also contain lipids. And they also contain bile salts, which aid in the digestion of fats. So we presented multiple fluids to represent the gastric and intestinal fluids, both a fasted and a fed state. Since food can impact the type and amounts of chemicals that can be absorbed, our approach is to do a fasted state versus fed state comparison in the testing to determine which one would be a worst case scenario and use that as our chosen biofluid for the study. We didn't present it here, but we also have found other intermediate stages of fed. So we have an early and also a late fed stage, which we will also test to determine which of these would present a worst case scenario for consideration as the selected biofluid for our study. And then now I'll pass the talk on to Ms. Marion Russell from the Lawrence Berkeley National Lab, and she'll talk more about the study setup for this.
Thanks, Jocelyn. Before I talk about how we propose to measure bioaccessibility, I'd like to expand on the working definition of bioaccessibility and how it relates to the process of biological absorption. Biological absorption is an active process, as illustrated in this model. This particular diagram is effectively a model of intestinal uptake, but I think it can be applied in a similar way to both the dermal and inhalation pathways. As the particles of crumb rubber pass through the system, chemicals are dissolved into the biofluid. These chemicals are then actively removed by the epithelial cell layer and passed into the bloodstream. The fugacity capacity of the biofluid will change as the chemicals are removed from the biofluid and pass through the cell membranes and into the body. This active process will allow more chemicals to remove from the crumb rubber, and a concentration gradient is created. So bioaccessibility is often defined as the amount of the chemical in an environmental medium that can be potentially taken up across a biological membrane during exposure. In this diagram, the biological membrane is represented in D, and on the left side of your screen, we're showing the biofluid, and the right side would be the uptake into the human body. When measuring a chemical's solubility in the biofluid, it's important to take into account the change in the fugacity capacity that is created by the action of the cell membranes. So we can see multiple equilibria are governing the bioaccessibility process in the biofluid compartment. It's labeled as transport medium on the screen. Process A shows the equilibrium of the chemical bound to the crumb rubber and its dissolution as a released chemical into the biofluid. There is competition here with processes B and C, where B, the released chemical, is being passed to or into the cell membrane, and C is the option where it could pass directly from the bound chemical, from the bound particle across the cell membrane. And the process of the desorption from the crumb rubber is also reversible. So effective measuring of the chemical's accessibility in this compartment will be governed by the process D, the uptake by the cell membrane, and it will change the equilibria process that are occurring in this biofluid. So even though we are measuring a chemical solubility outside the cell membrane plays a role of what the chemical solubility is in the biofluid, because as chemicals are leaving, it creates an opportunity or a concentration gradient for more chemicals to be dissolved into the biofluid. Yeah, I clicked too much. Yeah. We are proposing to use a solid phase extraction process called stir-bar sorptive extraction that will take into account the change in chemicals' apparent solubility due to the active process of biological absorption. This will be an improvement over the static incubation method, which is commonly used. Physiologically based extraction tests consist of shaking a sample with an artificial biofluid at a biologically relevant time and temperature. This process reaches a steady state once the media is saturated with chemicals. So there's nothing in this process that's changing the fugacity capacity of the biofluid. As you can see here, the traditional method is a multi-step process that also limits enrichment of the endpoint chemical. After the incubation period and filtering to remove crumb rubbers, the aqueous biofluid is then solvent exchanged to an organic phase. This exchange is often repeated multiple times, resulting in a large volume of the organic phase, which must be concentrated and dried before the analyses can be completed. And the concentration process itself can result in some significant losses, either degradation or loss. The chemicals can stick to the side of the flask and be lost. In contrast, the stir-bar sorptive method is simple and provides an extended dynamic range and a large enrichment factor. The stir-bar sorptive extraction method was first published in 1999, and it's been used extensively in environmental analyses, forensics, food, and life sciences. And the method does really well at capturing chemicals from dilute aqueous environments and has been previously used in biofluids such as urine and blood. The key to the method is the glass-coated magnetic stir bar that has a solid phase that provides essentially an infinite sink for chemical collection. So the method is relatively simple, and I have an example here. Extraction and enrichment are conducted in one step. So this example shows 1 gram of crumb rubber stirring at about 1,000 RPM. It provides a well-mixed solution. And in this example, this was at room temperature, but you could easily incubate this at 37 degrees Celsius or your desired temperature. So after the desired length of time for stirring, you simply remove the stir bar with a clean pair of forceps, rinse off any particles with DI water, pat it dry with a chem wipe, and the stir bar is ready for analysis. It can be directly analyzed by thermal desorption for a VOC analysis and gas chromatography mass spectroscopy, or in the top arrow, you can go to LCMS analysis by backolution with a solvent for instruments that require a liquid injection. So the stir bars come in a variety of sizes, as shown in the top diagram, and coating thicknesses to provide a range of capacity and options on the coating to allow for a wide range of chemical classes to be analyzed. So we are proposing using 2 types of coatings. One is the polydimethylsiloxane, or PDMS, which is effective for lipophilic chemicals. And this particular one can go to VOC analysis, semi-volatile analysis, as well as non-volatile analysis. The second one has a silicone stir bar, and it's effective in capturing polar chemicals such as phenols and esters, and this one can be used for LCMS analyses. The mass transfer process in SBSE, or the stir bar method, is improved over the traditional shake and incubate flask. And this diagram here is showing that the chemicals from the crumb rubber sample move into the artificial biofluid, and the coating on the PDMS coating in this example on the stir bar provides an infinite sink. So as the chemicals are sorbed to the coating on the stir bar, they are pulled out of the biofluid, allowing more chemicals from the crumb rubber to move into the biofluid. So recoveries are governed by the oxanol water distribution coefficients properties of the chemical. And so you can monitor your recoveries and you'll know your range of chemicals that you're able to detect. So the PDMS coating in particular does well for the more lipophilic chemicals that have a higher KOH, whereas the silicone phase will capture the polar ones with the lower KOH. Let's see. So to use the stir bar, we're going to take into account all the factors influencing bioaccessibility. And in this example is the oral pathway. The factors influencing are the residence time in each biological compartment, along with the pH, temperature, and salt composition of the biofluids that Jocelyn just described. So for the GI compartment, for the mouth, we're proposing about 5 minutes, the stomach about 2 hours, and for the intestine about 18 hours. Next, I have some simple flow charts showing how we propose to measure the bioaccessibility for each exposure pathway. So in the GI system, we have a multi-compartment analysis where we'll take the mass of crumb rubber in the biofluid, and we'll start with the artificial saliva and a stir bar. Stir for 5 minutes, and then you simply drain out the biofluid so you retain the same stir bar and the sample of crumb rubber. We'll add the gastric fluid shown in the second compartment. Stir for 2 hours, then that's drained and replaced with the artificial intestinal fluid for stirring for 18 hours. After that, the stir bar can be harvested and go direct to chemical analysis. So this can be compared to a single compartment analysis, so we can also look at each compartment individually. And then it can be repeated, as Jocelyn mentioned, for the fed state, the fasted state, or any combination in between as we look for the worst case scenario. In the dermal system, will be a little more straightforward. We'll just be mixing the crumb rubber with the artificial sweat and sebum, and we propose here a slightly lower temperature of 32 degrees Celsius before the stir bar goes to chemical analysis. Now, the addition of sebum will likely pose a technical challenge here because it may compete with the stir bar, and we may actually find that the sebum may not be necessary. So it's going to be something we'll have to research and look into. An inhalation pathway for the lung, obviously the size particles that we use for the sample will be very important. So we will need to isolate a specific range of crumb particles to use as our sample, and we propose stirring 37 degrees for 4 hours before analyses. Now I have just one illustrative example. So this is sort of a proof of concept. This is an extraction of just one sample of crumb rubber. And in this example, it was the 1 gram of crumb rubber. It was stirred into 30 mils of just deionized water with 10% methanol. And it was stirred for 1 hour at 37 degrees Celsius. And it's kind of an initial range finding experiment. We wanted to see what range of chemicals we could find and what relative amounts were here. So the stir bar was analyzed for VOCs by our thermal desorption process and GCMS analysis. The pie chart is showing the range of chemicals we found, and these are grouped by chemical class and just by the percent area that normalized to the GC response of the instrument. So we identified 72 chemicals, and over 50% of them were heterocyclic compounds that represent the chemical signature of rubber. We also found a large percentage of aromatics and polycyclic aromatics. Next, I actually have a list of all the 72 chemicals, so it's kind of a long slide. I apologize for that. But I just thought it'd be interesting. This is also a log scale, so the differences here are an order of magnitude. And starting at the top, we have styrene, which we found in relatively low amounts. And I should point out the red dashed line is what we approximately think our limit of quantitation is. So again, this is a relative amount by instrument response, so I can't tell you like how many nanograms this represents at this point. It's just an initial comparison to look at the relative size. So some of the more toxic ones we found were styrene, and a couple, this is essentially chlorinated toluene. So there were some halogenated compounds we found, and it was in relatively small percentage of the sample. So the list continues on the next slide. At the bottom was the chemical that represents the largest amount, and that was the benzothiazole. It's kind of the signature of crumb rubber that was also found in Dimitri's emissions experiment, as well as our thermal desorption. So it was something in common found there. And then we found quite a few PAHs, which I guess isn't too surprising. Pyrene. You can see their relative order here as naphthalene and fluorine. Okay. So thank you for your time, and I guess we're ready to open for discussion.
So are there any further suggestions about biofluid composition? I have a comment. Oh, go ahead, Debbie.
I have a question. I just had a question on the lung one. This is great, by the way. It looks like you guys really have done a lot on it. Why is it the 4 hours in the lung? How did you guys decide that time?
I believe it was from the literature. Is that what Jocelyn did, an extensive literature search? But we're open to suggestions.
I believe that's chosen as a range from the range of the clearance time of the particles, so the time they would be available to be engulfed by the particle.
So I wasn't sure if I wanted to bring this up, but you had included microbiota in the intestinal fluid. And, certainly there's a lot of attention being paid to the microbiome in multiple body orifices and tubes. It's obviously going to be different in different people, but there are some common features also. And it's not just the intestine. The oral cavity is, full of bacteria, as many of us know. And I think that probably does make a difference in terms of absorption of chemicals. Now, the question is, can you model it in a standardized way? And, it's way beyond my immediate ability to give you advice, but I do think you at least need to acknowledge when you're talking about the potential limitations of the model that variation in the microbiome across individuals could make a difference in terms of absorption of chemicals, because it'll make a difference in the pH in various compartments, even though the body's pretty good at maintaining pH in compartments. I just think, given that sort of the current view of in systems biology, human biology, is that we're a collection of cells that aren't just us, but all the microbiota that we have a commensal relationship with. So, again, I don't know if anybody has been able to sort of model microbiome variability in this kind of uptake of environmental chemicals, but I do think it's an issue.
Yeah. Well, one of the issues, and this is a bit, it's outside of my expertise, is I'm guessing the crumb rubber in the field has its own microbiome, and because people are sweating and spitting or whatever they do. And I know they put biocides, but we don't know how effective those are in killing all that off. So there's probably some microbiome there, and it's different from the microbiome that would be in dirt fields. And again, it's just an interesting, I don't know how much you can go into it, but it's an interesting side question, because it isn't just the microbiome inside of us, it's the environment. Like, we know this from residential environments. It isn't just your microbiome, your house has a microbiome, and that affects certain exposures you have. We don't understand it yet, but it's an interesting topic. I wouldn't leave out the microbiome of the field itself.
Thank you. This is Amy Kyle. What would you expect the variability in these phenomena to be generally in a human population, whether it's microbiome-ish or any other reason? I mean, is it a factor of 2 or 10 or 1,000? I'm asking just a big scale question, because I have no idea. Someone probably does, though. We don't know. Of these, going through these compartments, what are we calling them here? Yeah, compartments. Like, what is the variability among people, and how much of any given thing will move through one of these arrows? Is it 2 or 10 or 1,000 or 1,000,000?
Well, it's knowable. I mean, there are reports on the composition of human, physiological composition and digestion speeds and things like that.
So you're telling me to look it up myself? Is that what you're saying?
No, I know. Well, I mean, there's
Anybody have a guess? I'm just curious, because I don't know, and I'm curious what planet it's on. No one wants to even guess. Is John being too strict? Is that the problem here?
Well, I don't know whose hand up was first. Dr. Sheldon.
So I've got it.
Ed is a gentleman. I have known that for a long time.
I have a question, and it's sort of, I'm having trouble visualizing this. You said that you've got the crumb rubber and you've got something absorbed to it, and it rapidly goes into the water. These are organic chemicals. So you're telling me that the crumb rubber is less hydrophilic than the water itself? Because why wouldn't it want to stay on the crumb rubber? And then once it gets into the water, it goes on to something that I mean, I know it's an equilibrium thing. I mean, essentially all you're saying is that the stir bar has to be more hydrophilic than anything else, and the stuff in between really doesn't make a whole lot of difference. Well, that's in my own.
No, that's good. We're moving in the right direction as far as the thinking goes, because I have to say, my thinking is trying to stay as simple as possible and just put the elements in the system that are required to get to this. That's why we kept going back to, okay, this is our definition, and this is our working definition, and we kept going back to that. And so we want to get to that number that'll be useful for the risk assessment later.
Right.
And so as far as the answer to your question, it doesn't take very much solubility in water if there's absolutely nothing there to start with, for a chemical to want to move into that water phase.
And then it goes back to, then it is
But then it's
to
equally
nothing.
competitive
Yeah,
with that
okay.
with that sink, and so it's removed from the water phase. It may
Okay,
partition
okay.
back into the crumb,
Wow.
but ultimately the flow is going to be towards that stir bar, which is starting off at 0. It's starting off with a gradient, a fugacity gradient through the water into the stir bar that's fairly strong. Because if the stir bar starts off clean, the water starts off
I
clean.
agree with all of that, but then given that and what you said about adding the sebum, it almost sounds like adding all of this other stuff is just sort of, you don't know if it's relevant or not.
So here was my take on that. We came in after your first meeting,
Right.
and we heard the problem and started thinking it through and coming with a solution before we understood the suggestion. And my premise was that the suggestion came from the flask method, where if you
Right,
increase
yes.
the fugacity capacity in the flask method by
Right,
adding
right.
fats and lipids and wax, you're going to bias the answer up to where you think it might be relevant. But in the case of the method Marion described, if you have the stir bar there and you add fats and lipids and waxes, you're actually going to bias the answer down in that case, because some of the stuff's going to want to stay.
So we are agreeing that
with the
all
fat.
the other stuff added may not make a difference, other than
Makes it more real, but
anything else that might impact degradation or change of the chemical. And I thought that stomach fluid had various enzymes in it that could, change the status of the chemicals. And the stomach fluid did not have enzymes in it.
The only enzyme present was pepsin, which is the most important enzyme. The compositions, other ones we've seen, typically it's just pepsin that's present.
In the gastric fluid.
The
Okay,
largest, in the
okay.
largest amounts, yeah.
So before I recognize Ed, you were going exactly where we were supposed to go in terms of addressing one of the issues that folks had was, do we think, and I'm just going to say it more simply, that the stir bar method can achieve the same goal as what we were recommending in terms of adding lipids, at the last meeting, because it's actually, I think, simpler, as Randy said. So I don't know if people have comments there, but this.
I had one comment, and I think it's on this topic. So I think it's really important to focus on the question of bioavailability. And of course, the ideal way to get it is get thousands of people and do all kinds of measurements, but we can't do that. We have to do it in a laboratory. I mean, we can't, create, well, we could if we had enough money, but you could really get, artificial stomachs, grow stomachs in cell plates. But I think it's important to get at bioavailability. I also think you have an opportunity with only a slightly bit more work to report all the fugacity capacities of each of these fluids. And then at some later point, somebody could take the measured concentrations in crumb rubber, and then you could actually see what you're observing, and you could plot, fugacity of one medium against another in real measurements and see how it all lines up. I mean, it's just another way to take a tool to observe what you're seeing in a pattern, right? So all you have to do is make a few slight if you calculate the ability, you've already calculated the fugacity capacity. You just haven't put it in the right unit. So you can do that. And you'll just have one more like window about these substances for these different fluids. And I think somebody could get a lot of mileage out of that in a paper like someday by taking these results and analyzing, showing, oh, look at how they smile.
Oh, did somebody, did you?
Ed is still waiting. I'm still waiting. Linda.
Gentleman Ed, go.
Yeah. So I have a couple of questions following up on what Drs. Kyle and McCone said in terms of thinking about not only the representation or the representativeness of what you're doing, but the confidence with which the public and even the technical community will have in how you report it. And so, yes, no question that we'd like to do thousands of population, et cetera, and that's completely infeasible. We're not going to do that. So then the question turns on what kind of error bars or windows of confidence around the data that you're showing. And so it wasn't clear from the data that you showed, sort of from the crumb relative mass details, whether that was based on 1 sample, 10 samples, what the variability of that was, how reproducible that was if you did the same analysis 2 times in the lab. But I think it would help us to sort of have more confidence to know that was backed up by some sense of some error intervals. And that's just a comment. Maybe you're already going there, but the other issue, maybe of more interest to me, which is not exactly in the biofluids, but sort of up at the front end, is the inhalation parameters that you define to sort of get the whole thing going. And Jocelyn sort of started right there, but just sort of went right by it. So I wasn't sure, I might have missed it. And that is, from adolescence to adult, there's a wide range of deposition patterns, of airway architecture, of dimensions, ventilation rates when they're running around the field, et cetera. And so you're going to have to pick something or some range of some things to describe what the particle deposition and so forth are, where you go from there. And I'm sure you'll think about this and it'll be thoughtful, but I think you need to describe what that is in a way that, we feel like it's included the sort of the right range and that we're there because, we expect that we'll get good, useful information or interesting information in terms of the particle size measurements that will be made on the field. But depending on the ventilation rates and the size and structure of the lung, the deposition patterns may be somewhat different. And then when it gets to the lung fluids, and et cetera, then we can go from there.
But isn't that, just to push back a little bit, isn't that in the risk assessment where you deal with ventilation rates and varying structure of the respiratory tree in children versus adolescents?
Well, maybe, but if they're building a model that describes what the range of these values are going to be, they have to have some decisions about it.
Isn't the model more of what happens when material gets to these compartments as opposed to how much
gets there?
Yeah, I know.
I could take a chance at just trying to answer that. Yet, yes, I think the bioaccessibility term, not bioavailability, but bioaccessibility is just how much of this chemical, once it gets into this part of the body or this place, how much of the chemical in that product or material is accessible to the body. Some of it the body never sees. It'll go right on through, and that's what we want to know. And so, yeah, if you inhaled particles, some of it will go deep, some of it won't. Well, those that don't probably are going to get ingested. And so at some point they'll get in the body and they'll go through a different path. But still, you need to know of the...
Or coughed up.
Of the 100 units, or spit out on the field for the next kid to get on it. So yeah, but you need to get a sense of the relative amount that's in the material that could be accessible to the body. And that's what I think the effort that we're putting into is that going after that accessible number.
Along with what Ed was saying, it sounds to me like you have not compared the shake method to the mass transfer method. And you're asking us, do we believe that one is going to be like the other? My response is perhaps you should test the two of them to show me. And then, sort of, I mean, obviously the second one's easier. And is there a way that you can take that data and use that approach if they are different? And so that's what I would recommend.
So someone mentioned particle size, probably Ed. It certainly makes a difference in terms of deposition in the airways. But in terms of bioaccessibility studies, what size particles are appropriate? If they don't get enough, fine particles to study, can they use bigger ones? Does it make a difference?
Well, it may make a difference on what the surface chemistry is, and what absorption, et cetera, are taking place, and what exchange there is.
Yeah. That was one of the questions that we're supposed to be answering.
So you said that you were able to model the particles and what, sort of area they went into, and it sounded to me like you did PM10 and PM5, but you should be able to go down to much smaller levels of the crumb rubber, and it would be good to try to model that and sort of see, what's going to be there, which fractions you would expect to have most in the lung, and that would probably then drive what are the particle sizes that you need to do these kinds of experiments on.
I guess there are a couple of issues here. If we are talking about crumb rubber, then we can fractionate based on their size and find out different portions. On the other hand, if we are going to collect airborne particles, it would be not easy to collect sufficient amount of PM2.5 to do, extraction by biofluid.
I think it should be crumb rubber, but I think that, there's a lot of argument that it breaks up into little fine particles, and so if you can take it, sort of do some kind of, you run on it for 2 months and, then sieve it.
Yeah, what I'm trying to get at is that either way have certain limitations.
Right, yes.
I think it's always important to acknowledge those limitations, and you may not be able to get enough chrome rubber PM, like you say, in terms of ambient monitoring. You certainly can get plenty of crumb rubber. Yeah, exactly. Particles. But I understand the issue with regard to ambient monitoring.
But the issue is they can be different from
Yes.
airborne particles.
Yeah, no, I understand. Amy?
It seems like this is important, though, to figure out the little particles, because the air pollution people are saying that, the road rubber fraction of air pollution around freeways and roads is very harmful. And so whatever, it's that issue I think is being elevated, and I don't fully understand where we're figuring out the particle size distribution of these particles in these studies. Maybe we haven't gotten to that yet. But it seems like we ought to be able to respond to that in some way here. And I'm just not sure if we can. So maybe that's a question to those of you all who understand that issue a bit more than I do.
Well, as one of those air pollution people, I agree that road dust is definitely something that we are concerned about in terms of health impacts. And that's a combination of tire and other motor vehicle wear plus road wear. So it's a complex mixture. I think that the field study, the issue is not so much that they can't get chrome particles from the field, from the surface, and those will be different sizes, and they will find out, what those sizes are. But will they get enough PM to measure from the ambient monitoring? That's chrome rubber.
Yeah, and I understand that, but my issue is apart from this question of the beaker or the stir, where are we thinking about these particles overall, not just in this particular context? That's what I was trying to say. So I'm not sure about that.
I think particle size is an issue. And another air pollution guy down there is going to...
From another air pollution guy. So I'm not sure I understood the import of your comment there, Dr. Bones, because my understanding is you're not collecting this to regenerate it in the context of a study. You're doing some sampling of the particle size distribution in the field to get some idea what the particle size distribution is. But you're not planning on resuspending that collected material. So once they have that, and they know something about the proportionality of it.
Okay, well, I misunderstood David then.
Yeah, the issue is, would we collect in the airborne PM 2.5 in sufficient quantity to do a biofluid extraction and get meaningful results?
Well, so if that's the issue, I think you got to be careful. Because it's one thing to collect it because you can collect it. And it's another thing to collect it because it's what's representative of what the exposure is likely to be.
So is there a point where someone's collecting the particles that are representative of the exposure as a whole?
Later on in our section, we're going to talk about field
Okay.
sampling, and we have been and we are going to do particle collection and also size distribution on the field. And we're planning to do a different height. So more detail.
Okay.
Yeah.
That's good.
Any other questions or comments on this section? If not, we can take a short break. I have 15 minutes on the schedule. Yeah.
More like 18.
So do we need that much of a break or 10-minute break?